All right, so Armando asked me to talk about challenges in RNA splicing. So I'm a biologist in the biology department here, and really a computational biologist by training, and also director of the MIT's computational and systems biology program. So I was very happy to get involved in this project. So I want to just briefly sort of review some of the sort of biochemistry of splicing, which is sort of at the foundation of this, since there is a biochemical process that we're attempting to model computationally here, and then kind of draw out some of the computational challenges. And so this model shown here is this clay model is actually a picture of splicing with the RNA transcript shown in yellow, the RNA polymerase shown in blue, and it emphasizes that splicing is actually occurring co-transcriptionally in the process of transcription, which is an interesting aspect of the problem. All right, so we've learned from deep sequencing in the past few years that almost all human genes undergo alternative splicing, so that there's just lots and lots of splicing. So almost all genes must undergo splicing in order to produce the correct mRNA. And then there's this opportunity for variation in that splicing process, which can occur in different cell types or tissues or different conditions here. So splicing just briefly occurs in the nucleus, and then involves removal of introns and ligation of exons, as Armando introduced before, producing a mature mRNA that is then exported to the cytoplasm where it directs translation. And you can, the process of alternative splicing is where you use different combinations of these exons. So for example, to produce these two different mRNAs, the longer one on the left with a pink exon and the shorter one that lacks this exon. And these mRNAs will typically encode different proteins, although sometimes they may differ in other aspects as well. So just to briefly introduce the chemistry, splicing involves two chemical steps. In the first step, a segment near the 3' or downstream end of the intron forms this unusual branch structure where it forms a covalent attachment to the 5' end of the intron here. Here the boxes represent exons, the lines represent introns, so you get this branched RNA. And then in the second step of splicing, the two exons are joined together and the intron is released in this so-called lariat form. The intron is typically rapidly degraded, and then the exons go on to become the mature mRNA. So quite a bit is known about the biochemistry of splicing. I just want to kind of briefly introduce some of the players here. So splicing is catalyzed by a very large protein RNA complex consisting of several small RNAs and literally hundreds of proteins called the spliceosome. It is not a pre-built complex, but actually assembles dynamically on the primary RNA transcript. So there are these components called SNRPs, U1, U2, U4, U5, U6, which are the key RNA protein complexes. So the U1 SNRP recognizes what's called the 5' splice site at the 5' end of the intron. And then there are components, U2AF and others that recognize signals at the 3' or downstream end of the intron. And these components replace one another. So this BBP and U2AF that are there early are then replaced by U2 prior to the first step in splicing in different well-defined complexes. Again, the details are not critical here, but I want to give a sort of an overall picture of the chemistry that's happening. And then this component U1 at the 5' splice site is also replaced by another component U6 prior to formation of this catalytically active complex here. Here you have the cleavage of this junction and the ligation onto the so-called branch site adenosine there. And then in the second step, the two exons are removed and the spliceosome remains attached to the lariat, and then the lariat is degraded and the spliceosome components are then recycled. And Recently, with advances in cryo-EM, we actually have structures of a number of these components, the SNRPs, and even whole spliceosomes, which is one type of data that that could be brought to bear in this project. Okay, so where is the information for splicing? So some of it is clearly in these sequence motifs that occur at the boundaries of exons and introns. So here, these data are for human. Here's a picture of the 5' splice site motif, which occurs here, is recognized by U1. The 3' splice site motif is kind of a bipartite motif with a pyrimidine tract here, composed of Ys or pyrimidine nucleotides that are C or T. And then this short YAG motif here. And then there's this branch point motif, but as you can see, it's quite degenerate. It doesn't contain a lot of information. But these motifs, at least in human splicing, are clearly not sufficient to specify the boundaries of introns. If you make models of these, try to simulate splicing with these alone, it doesn't work well at all. So clearly there's additional information present in the transcript that's used by the splicing machinery, and that's really a big challenge of this project. And so molecular biologists have described different types of elements that exist in primary transcripts that contribute to splicing. Generally, we call them splicing regulatory elements, sometimes SREs, and have generally categorized them into 4 different types. So they can exist in the introns and be intronic splicing enhancers or silencers, or can exist in the exons and be exonic splicing enhancers or silencers, where the enhancers promote inclusion of the exon and the silencers generally block inclusion of the exon or block splicing. And so these elements contribute to alternative splicing and are involved in decisions about production of different isoforms in different tissues, but they also play a role just in the basic splicing of a constitutive splicing as well. So these elements, these splicing enhancers and silencers, are recognized in general by proteins. So just as the core splice sites are recognized by the SNRPs, these enhancer and silencers are recognized by proteins in the HNRNP family, the SR protein family, which are ubiquitously expressed in all cell types, as well as by tissue-specific factors, including those from the RB-FOX family, the self and muscleblind family, and literally dozens of others. And exactly how they assemble onto the transcript and influence one another's interactions and spliceosome assembly is not well understood. Okay, but we do know of particular interactions that are definitely important in splicing. So in particular, these SR proteins recognize these exonic splicing enhancers and then interact with components of the core splicing machinery, such as U1 SNRP and U2AF, as shown here, to promote recognition of that segment as an exon. So human genes have a very expanded geometry compared to the genes you might have seen, genes in bacteria, or the typical representations in textbooks. And so I just wanted to emphasize that here, that the mean length of the primary transcript prior to splicing of a human gene is at least 30,000 bases long. They're typically 8 to 10 or more exons. The exons are much, much smaller than the introns. So here, below here, I've drawn this to scale, and you can see the exons are about 150, the introns 10 to 20 times as big. Basically the lack of clarity on exactly where the information is in the transcript that the splicing machinery uses leads to a number of interesting computational challenges. And I've broken them into sort of 3 challenges here, which I'll describe: the constitutive splicing code, the splicing phenotypes of genetic variants, which has a very practical application, and the alternative splicing code. And I should emphasize that all of these codes have lots of interesting practical applications. Okay. So The constitutive splicing code is sort of the simplest to describe. It is to predict the predominant splicing pattern of a human primary transcript using the same information used by the splicing machinery. So we're given a sequence of length, roughly 30,000 bases long of A's, C's, G's, and T's. And then we want to predict the coordinates of all these exons. So the first exon starts at base 1, but where does it end? Where does the second exon begin and end, and so forth. And it has to be to nucleotide precision, because being off by even a single base will throw off the reading frame and potentially produce the wrong proteins. And so, of course, this assumes that the gene produces a single or predominant mRNA isoform in all cells, which is true for some genes. Okay, but very importantly, the algorithms that are developed in this project, it is sort of our goal here is that they should involve interpretable components and rules when possible. So they should involve particular motifs that have a function at the RNA level, perhaps being recognized by particular protein factors. That factor should be represented in some form in the algorithm. There might be combinatorial effects. If you have this factor plus this factor, then it recruits a U1 SNRP, whatever. There could be distance constraints and so forth. But all the elements in these algorithms should relate in either directly or indirectly to actual biochemical components of this process. And I think that's really what's so unique about this particular effort here. And so this is true of this constitutive splicing code, but also of the other codes. So besides human, there are also constitutive splicing codes for other organisms. Introns tend to be smaller in other organisms, so that's why I've drawn a different geometry. So there's some differences in the rules between animals, plants, and fungi, and so that creates a whole set of related problems of varying degrees of difficulty. Again, each of which could provide insights into mechanism and could also provide have practical applications. So what kind of data do we have available? So, of course, we have the human genome sequence. There are roughly 20,000 protein coding genes in the human genome. For most of them, a predominant splicing pattern can be identified. And so we can construct data sets for training and testing of algorithms consisting of, on the order of 10,000 genes. The genes vary in size from about 1,000 bases to 100,000 or more. Again, about a dozen exons per gene, roughly. And so we can sort of make a training set that has 100,000 exons and introns and similarly sized test sets. We have made such data sets for this project and have also been making data sets for other organisms, which are also being explored. So the second major challenge I'd like to highlight is the prediction of splicing phenotypes of genetic variants. So rather than sort of predicting where the exons are de novo, you may be given a particular, let's say exon 3 of this particular gene, and there's a known genetic variant, a position in the genome where people differ from one another, either patients with a particular disease or just natural variation in the population. So perhaps some people have a GTAAGC here at this 5' splice site, and then a rare allele is to have a T at that position, +5 here, shown here in red. And the question would then be, if you look at the motif for the 5' splice site, you can see that the G is preferred and the T is not preferred. So therefore, this variant would be predicted to weaken that splice site. But does it matter? Maybe this exon is sort of strong enough that it can tolerate that variation, or maybe it will completely disrupt splicing. And so that's sort of the challenge here, is to predict, will this variant cause this exon to be skipped by the splicing machinery, or spliced normally, or perhaps some other phenotype, some other splicing pattern might arise. So the NOMAD consortium, involving people at Broad and elsewhere, has sequenced 70,000 high-quality human genomes. And so these contain very large numbers of human genetic variants, 550 to 100 million common and similar numbers of rare variants. And so we have lots and lots of variants to work with. There's somewhat less data available where we actually know what the splicing pattern is, but there are lots of efforts to do RNA-seq on different cells and tissues of people with known genetic variants. And so there's starting to be more and more data available where we know the splicing pattern of both variants. And one study detected about 3,000 splicing quantitative trait loci, or SQTLs. And so there are starting to be sets of examples of variants that do affect splicing. There's also many sort of disease-specific data sets with typically hundreds or thousands of variants that are associated with disease, often by interfering with splicing. And so I just want to point out here that the most accurate available methods, both for this sort of constitutive splicing code and for variant prediction, use deep neural nets, like in particular this Jagannathan paper here, but they're not interpretable, and they don't really correspond to an underlying splicing mechanism in a way we can understand. Okay, so finally, alternative splicing is particularly interesting because of all the different alternative variants that it can produce from a particular gene. A single human gene can, in principle, produce dozens, hundreds, or even thousands of different mRNA and protein isoforms. And so there's lots of different types of alternative splicing, at least 7 or 8 that are shown here. The most common is this cassette alternative exon splicing shown at the top, where you either include or exclude the blue exon. But you can also have variants where you include a longer or shorter version of an exon, also either at the 5' splice site or at the 3' splice site, as shown here. You can sometimes retain an intron in the mature mRNA, and there are more complicated patterns in which you have mutually exclusive inclusion of 2 or more different alternative exons. These types down here also are quite common, but they involve some components that are a bit outside of splicing. They involve alternative promoters or alternative cleavage and polymorylation, and so that doesn't fit within the scope of this project as currently conceived, but could be an interesting follow-up investigation. Okay, so the alternative splicing code, just to sort of define it more precisely, is to predict the set of alternative mRNA isoforms expressed by a gene and their relative abundance, which we often in the field call psi values for percent spliced in values. So what percentage of the time is the red exon included in mRNAs, for example, or if there are 3 different isoforms, what is the relative abundance of each of those 3 isoforms? And again, that can vary for different cell and tissue types or developmental stages or conditions and so forth. Fortunately, we have a lot of good high-quality RNA-seq data from different cells and tissues, so we can actually measure these things reasonably accurately. And you can think of 2 different versions of this alternative splicing code problem. You can predict the isoforms from scratch, so just, again, like the constitutive code, just start from primary transcript and predict those isoforms. Or perhaps we could think of an easier version where you're given the set of isoforms, maybe we discover them by RNA-seq, and the challenge is to predict their relative abundance, or which ones will tend to increase in abundance in one cell type or another, or in disease versus health, and so forth. And there's some nice work from Brendan Fry and others at the University of Toronto on this problem. So examples of differential alternative splicing include predicting variants that differ, or exons and isoforms that differ between different tissues, such as brain and liver, between different disease states, such as cancer versus normal. Splicing changes a lot during development. That's also interesting. There's even differences in splicing between in male and female, for example. So what data do we have available? So the GTEx consortium has produced lots of high quality RNA-seq data from human tissues that we can use for this project. Can estimate mRNA isoform levels for most genes. And there's also lots of data from ENCODE and other sources. I just want to finish by just mentioning there's also quite a bit of information from the recent ENCODE phase 3 project that focused on RNA binding proteins that my lab was involved in, together with Jean Yo, Brent Gravely, Eric Lecrier, and Jiandong Fu. And these are different types of data that are often related to RNA splicing. So we've done in vitro studies of splicing proteins, what motifs do they bind in vitro. Jean Yo's lab has found where they bind in the transcriptome using a cross-linking approach. Brent Gravely's lab has done a knockdown approach where it interferes with the function of the protein and then looks to see what splicing changes by RNA-seq and so forth. And so these data can also enrich some of these models. So we're looking forward to continuing to collaborate with this talented group. I'll stop there and be happy to take questions.